Okay, good morning, class. How are you today? How's your health? So, importante kaayo
Morning,
no
ma'am.
nga kanang maayo atong lawas. We are okay. Very good. So, please continue to be safe at home because the COVID cases are still um, rising, and we don't want you to be infected. And also, um, naabot I don't think is it okay na na vaccinated na ba mo tanan, or kay mga estudyante man pud
Yes,
usually
ma'am.
na vaccinate na mo.
Na-vaccinated na ko, ma'am. Sinovac.
Very
Ako pod, ma'am.
Oh, how what brand of John Celestine?
Tupak.
Sinovac pod. Si Krista, Abi May,
First dose pa
Krista,
lang,
Abi
ma'am.
May.
Ay,
Ay, sorry.
Ma'am,
Vaccinated na pud ka, dear.
na-check pa ko, ma'am. Next siguro.
Okay. How about you, Krista Rosette?
first dose pa lang, ma'am.
First dose pa. Oh, it's okay. So, ako pag vaccinated na po. So, that's good for you that you're already vaccinated, no? So, if kanang magipit niya, let's encourage our friends, our families to get vaccinated. So, you can really share some information because it's more on biotechnology nga aspects sa vaccination. So, please do encourage your family and friends to get vaccinated because um, this is really the major solution. Not not really the only solution, but this is a significant solution to the pandemic, to this SARS-CoV-2 virus pandemic. So that's good. That's really, I'm glad that majority of you are vaccinated. And so today I will be discussing the exercise 3. So tapos na to sa 1, then 2. So I hope that you are already starting your accomplishing your lab reports so that you can really submit it before the deadline or within the deadline. Okay, so for this exercise 3, so I will be discussing and you will be the one who will do the experiment at home. Okay, so this is called an experiment at home. So later I will be discussing the procedure, but it's just simple and the materials are just available at home lang. So I modified a little bit last time so that it can really fit in sa uh, home setting. Okay, so before I will proceed, I will also would like to announce that I will give a quiz for the exercise 1 and 2, and I will post it on the VSUEE uh, at 12 noon. Okay, so I will chat in the group chat so that you will be notified and you have at least 24 hours to answer the quiz. Okay, so it's just in the VSUEE, so I will just set it sa iyang ang time frame to for you to answer. Okay, so please study the exercises 1 and 2. Okay, I will now proceed to the discussion. I will now present my screen. Your Okay, this is the laboratory part and this is exercise 3. So the title of your exercise is determining threshold limits, test for salt solution. Okay, so before I proceed, may I ask if have you started sa lecture na part? Where are you now sa inyong lecture, dear? Nag-start na ba mo class?
Start na, ma'am.
Okay. So, nag-start na mo. So, in what part na mo napit? So that I can adjust in because most of these principle are ano naman sa lecture. History. So, history niya siguro mo.
Oo, ma'am. History, ma'am.
Diretso, ma'am.
Okay. Okay. So, most of these principles in the threshold limits, mga those response relationships, you will get along this in your lecture class. Okay, so because it will be the first time that you will encounter these words, but I will just explain it to you. So, may I ask every one of you, class, if what have you eaten for your breakfast this morning? So, or nakakaon na bumutanan or wala pa mo kakaon?
Ano,
Wala
ma'am?
pa.
Wala. Oh, or you just woke up. Ayan. Okay, so what have you eaten this morning?
Rice, ma'am. O kanang maling.
Maling. Wow, great breakfast. How about the others?
Wala pa ko kapamahaw, ma'am.
Diretso.
Okay, so after the class, or you can, ano naman, you can eat while listening to the lecture. Nga, I can just permit you to do that. Anyway, so thank you for um, sharing what you have you eaten this breakfast for your breakfast. So I asked that from you because this morning we will tackle all about the neural taste or our taste neural system because it will relate to the exercise, which is the test for a salt solution, because we will use our tongue, or the members of your household will use your tongue for the indicators of the threshold limits. Okay, so I know that uh, a diversity of our age group, so the young or the old, have different preference with in terms of taste. Okay, so we have five distinct tastes, and mostly children wants to have a sweet food, wants to that their food is really sweet, so that um, and it will really make them more energetic. And also, if we also eat cakes, we also eat candies and other foods that are really sweet, of course we tend to be energetic because it's an energy source, diba? So some of us have a sweet tooth, and other people prefers to have a sour taste. Okay, so these are these belongs to the five distinct tastes that our tongue can really perceive or can really detect. Okay, so third naman is we have a bitter taste. Okay, so it could be that it has potential poisons, it could be like that, but mostly when we eat vegetables, when we eat internal um, organs, cooked internal organs from chicken, from um, pork, so or pigs, ganun. So we can really taste can really detect bitter taste because some of them contains mga polyphenols, it has tangy taste, ganon. But usually it has potential poison. That's the connotation or significance. And the fourth taste is that um, our tongue can detect salty taste. So essentially, the sodium is really the um, essential to physiological processes. So this sodium or mga salts are really important in the these are majority of them are cofactors in the mechanical metabolism. Yes.
May nag-ask to join, ma'am.
Okay, sorry. Sige, there. Thank you. Because, okay, thank you. Please notify me kay Murag isa di ko ka kuan. Okay. Okay, so in the fourth, again, the fourth distinct taste is the saltiness. So sodium is essential to physiological processes because these are cofactors in the of most of our enzymes in order um, to function in the biochemical mechanism in our muscle, in our blood, so in our cells, inside the cells. So that's really if essential or important in the physiological processes. Okay, wait. Okay. So, and the fifth taste is the umami taste. No, so these are uh, proteins. It can proteins that is detected by our tongue. So it really umami taste. Proteins to grow and repair tissue. So that is why majority of kanang naa sa pork, sa chicken, they have umami taste because it has lots uh, of proteins there. Okay? So yun ang five distinct taste. Okay, so now let us uh, get to know or understand our tongue. Okay, so in our tongue class, so if we examine our tongue, we observe that there are bumps in our tongue and it is called papillae. So these are papillae. So this papillae help grip food with your teeth in chewing. So it will help you in holding the food so that it will have in grinding the food that you eat by your teeth. Ganon. So it is also another special job. So in that papillae, it contains 200 or more taste buds. Okay, um, so in that, as you can observe in your tongue, no, here in the picture also, there are bumps, and in that, the bumps are called papillae, and this papillae contains 200 or more taste buds. Okay, so these are the um, the part of the tongue that where your taste bud is located, or it can really the specific location that the taste you will detect the taste of your food. Okay, so individuals vary in their sensitivity to taste sensations. Now, a function of density of these papillae on the tongue. Okay, so we have different numbers of papillae in our tongue. So there are really people who have more papillae than me. There are less papillae. There are people who have less papillae also, depending on the age, depending also on the lifestyle. So ganon. So that is why if you are familiar with no sa food technology that iba because they are uh, always conduct food processing ganun, so and then they have to undergo it in a sensory evaluation. So this is really important in really in the food technology side because they will really choose panelists. Actually, the technicality is that they are really trained panelists because they have to detect the different taste in your tongue because so that they will get they will be familiar with the taste. Good, so they will be trained in their taste buds so that they can really detect different distinct tastes and really can really analyze in the when they are now subjecting to sensory evaluation. So It really different people differs the density of this papilla on the tongue. No, it depends on age. It depends also in on their lifestyle. Okay, so now the papillae contains the taste buds. The taste buds, the function of this is that it really contains a pore that catches food. So this pore is the receptor cells. Okay, so sa kanang bump cell papillae, and then the papillae contains the taste buds, and the taste bud contains the receptor cells, in which that these taste cells will have receptors, in which these are more antigen antibody nga kanang um, antigen antibody linkages, ganun. So it is taste cell. There are different taste cell that these sweet compounds or sweet sugars or sour compounds or chemicals will bind to this taste cell receptor cell, and it will then detect, it will then send signal to the brain and it will really recognize what taste uh, what food what is the taste of the food. Okay, so each taste bud pore has 50 to 100 taste receptor cells with antenna like hairs and sense food molecules, diba? So that's really how amazing the tongue is. So, ganun pala how it works, no? In our tongue when we eat food. We can really determine, di ba, with sometimes we don't like the taste of the food. So, we prefer another restaurant because we don't like the taste. So, ganun. So, it's because of this receptor cells that are located in the taste bud that will sense or detect, that will really link to the receptor taste cell and sends a signal to the brain and recognize what is the taste of the food. Okay, the simplified diagram shows the general idea that we experience specific taste when a food molecule fits like a key. Parang lock and key model sa enzymes food, kanang sa mga inhibition. Um, when a food molecule fits like a key onto a specific taste receptor, triggering a signal to our brain. So genes determine the sensitivity of taste cells. So there is still a factor on the genetic side to our sensitivity of the taste cells. So it also differs no sa ato at taste buds, the number of the taste receptor cells. As you can see, as we know that we have 50 to 100 taste receptor cells. So each a person differs the number of the taste receptor cells in each taste buds.
Isis mi ma'am na ay mag-joining ma'am duha pa ka buok.
Okay. Wait. Thank you. Okay. Sige. Okay, I will continue. So to have a clearer view, to have an in-depth understanding of our tongue, as you can see, this is the part. These are the parts of the tongue. So as you can see, no, um, here at the tip of our tongue, here is a fungiform papillae. Okay, there is the fungiform papillae, and at the sides of our tongue, there is a foliate papillae. Okay, so um, um, at the middle and at the end of our tongue, we really have circumvallate papillae. Okay, and it really differs the sensitivity of the taste. As you can see here in the graph, there you have the taste strength. Okay, so all parts of the tongue taste the saltiness, sweetness, sourness, and bitterness, no? So, but it really differs on the taste strength because of the numbers of papillae or in the numbers of cell receptors inside the taste buds. So in the diagram A, as you can see here, this is the tongue. So the taste buds line the trenches around tiny bumps in the on the tongue called papillae. Okay, there are three types of papillae, which are distributed on the tongue as shown. Okay, as I have already um, said that to you, and the taste buds found in each of papillae show slightly different sensitivities to the four basic tests as mapped out in the graph at the top. Okay, so the sensitivity of the different papillae varies across the tongue. So as you can see here in the graph, so maybe the most sensitive papillae is located on the um, middle or the end of the middle end of the tongue, in which here is the circumvallate papillates, but the sensitivity of this circumvallate papillate to saltiness is low, while here in the folate papillae, so here at the sides of your tongue, it really has a high sensitivity to saltiness. So, okay, so it has just for the bitterness bitter taste, uh, it is relative that they have the same sensitivity for the circumvallate and the folate papillae. But here for the fungiform papillae, it has lesser sensitivity or lesser taste strength than the other two papillae. So that is why, diba, you have to feel or you have to really chew the food so that you can really determine or detect the real taste of the food, diba? So you have to chew it thoroughly, and then so that um, those receptors or those compound of the food compounds will really be detected by this circumvallate papillae and also the foliate papillae. Okay, so the sensitivity to the primary taste varies across the tongue, but these variations are small, and all four primary tastes can be detected wherever there are taste receptors. So these are just small variations, but they will really detect all four primary taste as long as there are taste receptors. Okay, so that's the amazing amazing function of the tongue. Okay, so here is the more detailed illustration of the papillae, the taste buds, and these cells. Okay, the tongue is covered with bumps called papillae. As you can see, this is a papillae, and each papillae has taste buds. There are many taste buds in each papillae. So in this taste buds, there are gustatory cells. So the specific cell that are located in the taste buds are gustatory cells, okay, the cells that do the tasting. The tip of each gustatory cell protrudes through a pore in the surface of the tongue. So in the gustatory cells, there are nerves that are attached to it, and these nerves carry signals from the gustatory cells to the brain. The tip of the gustatory cells is covered with an assortment of bitter taste receptors which can detect a wide variety of compounds. Stimulation of any of these receptors sends a signal to the brain, and it will say that it is bitter. Okay, so as with other senses, um, um, your receptor cells influence your brain's response. So di ba? We have a something pre-assumption that someone will tell you that okay, their food is not that good, or it's not that tasty. So of course, so you have heard that, and there is now um a link to your brain that it says that okay, so maybe it really tastes, the food is really it is really uh it really tastes they taste bad, no? So there is now a bias in your brain. So wala pa gani mo na na-taste ang ilang food in that particular restaurant. You have already conditioned your mind that okay, it's really their taste is really bad. So that is why you don't like to ano na when you really taste it na for real. So there's now a bias in the brain that really says that it's really bad. Okay, and then likewise, being told that a wine costs $90 rather than its real $10 price makes an inexpensive wine taste better and triggers more activity in a brain area that responds to pleasant experiences. So it really there is really a biases there in when we apply it in the marketing side, no, because it's a something that you are ano, you really have that pre conditioned mind setting of that. So that is why, so it's uh, when the wine is $90, when you say that it's really $90, of course you have to because diba, as the wine ages, it will become more um kanang tasty and it really becomes sweet. And also as the wine ages also, so there's also an increasing of price. Now when we label this wine as $90, of course, but the real price is really only $10. So you just there's now the precondition of the mind that oh na okay, so this wine is has a good taste because of this uh, uh, of these costs that are really labeled here. So that is there is also a bias there. There is when it comes to marketing side, no. So that is your brain can be deceived sometimes when it term in when it is in terms of food, diba? And other also senses will also influence the taste of your food. Okay, so sometimes you, if you don't like the color of the food, you don't like to taste it because and it really sends a signal of it's really bad because you don't like the color or the presentation of the food. So that's there are a lot of uh factors that can really affect your taste preferences, no? And of course, class karon, ano, so there is also the placebo effect. The brain's thinking frontal lobes offer information that other brain regions act upon. So in this times of pandemic, so it is really important that we have a sense of taste. Kinahanglan dili gid ta ma-loss of taste because you know when we experience loss of taste and loss of smell, of course, maybe we can really these are really indicators of kanang if the RT-PCR, if you have not undergo RT-PCR, ganon. So the same one of the symptoms is loss of um, taste and loss of smell. So, of course, you will, maybe you are suspected for COVID if ganun ang nangyayari. So, importante karon yun nga naagid tay sense of taste and sense of smell. So, paniguruag yun. Maniguro gid ta nga natay sense of taste and smell. And it really, why is it that if we are infected with SARS-CoV-2 virus, so we tend to have a loss of taste and smell because of course the SARS-CoV-2 will bind to the nasal area, the throat area, and also to the lungs. So that is why they are really when there is infection, of course, our maybe the signals or the taste buds will not really function because of this infection that are already in our throat, in our nasal, in our throat, and in our, if it will really go down deeper to the lungs. So that is why there is now a loss of taste and smell. So yon, so ang importante karon na atay taste of smell, sense of taste and sense of smell. Okay, so our neural system for taste is remarkably sensitive. Okay, so our tongue plays a significant role in this experiment because the taste shall be tested to determine the threshold limits to the salt solution. And this exercise aims to understand the scope of threshold limits and be aware of our taste preferences on the salt solution. So this exercise also predicts what usual foods we eat based upon the result of this exercise. So this is now the before I proceed to the method, so I will explain to you what is threshold limits, and you will eventually discuss this further in your lab report. The threshold limits, it is the airborne concentration or the liquid concentration of the chemical expressed in ppm. So this in this limit value, there is no adverse effects. So there is no adverse effects that are observed in workers exposed for such particular chemicals. Now 8 hours per day in 5 days. So these are the limit value or the concentration that there are no toxicity or there are no symptoms that occur during the exposure and this particular concentration. Now, the TLV is usually set to avoid minor toxic effects. Now, like skin or eye irritation. So, of course, even though these toxic effects are just minor, so it should be identified. It should be set um, with the experiment so that you will really have a understanding of you will not limit your concentration when you are exposed to this particular chemical or this particular food. For example, you have an allergy. So in what concentration that you really can observe symptoms nga mo-occur na ang skin irritation ninyo, 'di ba? So that's the threshold limit value. So here naman diri sa atong exercise. So okay, another definition of this threshold limit is the concentration of the substance in air or in a particular liquid that can be breathed for 5 consecutive 8-hour work days or 40-hour work week by most people without adverse effect. Okay, so the learning outcomes of this exercise is to establish causality that the chemical has induced that observed effects. And second is to establish the lowest dose where an induced effect occurs. So later, I will discuss with you for this salt solution experiment. The third naman is to determine the rate at which injury builds up the slope for the dose response. So it is expected that in the discussion, in the results and discussion of your lab report, you will present a graph. Okay? You'll present a slope for the dose response relationship in the salt solution test. Okay, so this is now the method for the experiment at home. So class, the materials that you will be using, these are just found at home. So you will not have a hard time purchasing this one because it's really available. So number one, you have to prepare different concentration of salt solutions. Okay, since you have no analytical balance sa inyong balay, so you just have to dissolve 1 pinch of salt to 300 ml drinking water. So I know that you have a kanang sa mga bottled water gani, 'di ba? May mga 250 ml, may mga 500 ml, and I think that you have also mga calibrated nga mga bottles there or mga cups. So use it in order to measure the 300 ml volume of water. So this is for the 1000 ppm. So these are the different concentration for the salt solutions: 1000, 2000, 3000, 4000, and 5000. So for the 2000 ppm, you have to dissolve 2 pinches of salt to 300 ml drinking water. So ang imo lang kalahian ani nga imong i-measure ang kaning salt because kanang constant man ang volume sa 300 ml drinking water. Okay? So, for the 3000 ppm, you have to dissolve 3 pinches of salt and mix it in the 300 ml drinking water. For the 4000 ppm, dissolve 4 pinches of salt. And then for the 5000 ppm, you have to dissolve 5 pinches of salt to 300 ml drinking water. So, these are prepared individually. Okay, kinang naamoy kuan pwede ninyo nga kanabang sa mga pitchill, ingana. So, kana siya nga mga preparation. So, you will do it uh, the same day yun. So, and then, second, you have to inform your household members for this that you have an experiment at home so that they will cooperate and um, they will be tasting different concentration of salts. Okay, so the second procedure for this is to taste the salt solutions with 2 to 3 drops, okay, by each member of your household in ascending order. So mag-start mo og ipa-taste ninyo ang salt solution sa 1000 ppm, then after 1000, 2000, and up to the last concentration, which is the highest concentration, the 5000 ppm. So you have to record the concentration at which a person begin to taste the salt in the bar chart. So for example, sa ako okay, so if I have already tasted the saltiness sa 1000 ppm, of course, I will now so I will record it na there's one person that tastes the 1000 ppm. So um sa uban nimo mga kauban sa inyong balay, so another food that you will record the concentration in which a person will begin to taste the salt, no? So and for the number of your household, so it is better that you have mga 3 to 4 member of your household at different ages. Okay lang yan. Bisag imo pa mga manghod, mga 10 years old, ganon. So, it's a good thing that you have at different ages because you will really determine, you will really see the result that each has different taste preferences and taste sensitivities, no? Because as what I have discussed from you earlier, that if you are getting old, you have less number of taste buds or less number of taste receptor cells that you can really detect such specific taste. Okay, so it's really okay. So I won't give you a specific number because it depends upon the number of your household at home, no? So mas maayo nga pod nga 3 to 4 if you have less than 3 members in your household. So much better siguro if you can just um, uh, request your neighbors or your cousins, ganon. So 3 or more nga mga members of your household there. Okay, well really you will request them to participate on your experiment. Okay, so is it clear, class, for the method?
Yes, ma'am.
Okay, so And that's the whole procedure of your experiment at home. Okay, so for the discussion, you will present a bar chart, class. Bar chart of the result of this different concentration of the responses of your the taste sensitivity from the respondents. The respondents are your member of your household. Okay, so for the discussion, of course, as what I have said to you, using a graphical presentation, you have to present it and then provide explanation. Why? These are the results that has come out. Okay, the outcome of your result. Okay, and then provide explanation and provide such references of your explanation so that there will be more um, solid um, fact or that it is really relatable to this study that has been conducted also. So, ganon. So, yun ang iyong laman or that's the content of your discussion. Okay? Next, for the guide questions, so these are the guide questions that you have to answer also aside from the discussion. So, number one, if the point at which you began to taste the salt indicated the point at which death occurs, so it has to relate with the salt solution. Now, what if you began to taste the salt? It will indicate the point at which death occurs. So these are mga hypothetical questions. From this toxic substance, how does it relate to the following terms? So you will now define what is acute toxicity, B, what is threshold limit, as what I already discussed it with you. So C, what is lethal dose? How does it relate to the following terms? So lethal dose 50. And terms, these terms class, you will encounter this in your lecture part. Okay? Okay? And then second question. So if the point at which you began to taste the salt indicated the point at which a health effect occurred. So dili na siya death occur, but then there is now a health effect occurred from this toxic substance. How does it relate to the following terms? So, letter A, what is threshold limit? How does it relate to threshold limit? Letter B, individual susceptibility. You have to take a look of this definition and relate it to the result of your test, salt test solution. And then third is the dose response. Okay. Now the third question is, would tasting the water be an excellent way to determine the amount of salt in our drinking water? Okay. So if it is yes, provide an explanation. If it is no, provide an explanation. Now for the fourth question, if we cannot taste salt in water, okay, does that mean there are none present? So if it is yes, provide an explanation. If it is no, so still you should provide an explanation at this point. Okay? So that's the last question. So for these are just 4 questions class that you have to answer. And just ask questions along the way that you will conduct your experiment. So you don't hesitate to ask your instructor, ask me to how to clarify something about the experiment and also for the discussion. So I know that there will be more single inquiries that you will be asking me. Just don't hesitate to chat. Okay. So for the submission class, you have 2 weeks to conduct the experiment. So from this time, September 21 to October 5. Now after October 5, okay, you have 2 weeks to accomplish your lab report. So the deadline of submission is October 19. I will set a deadline there in the VSUEE so that you will be reminded that you will now submit it. Okay, so that's really for the submission part. So class, is it clear?
Yes, ma'am.
Yes, ma'am.
Do you have any questions or clarifications, class? So if there are no clarifications, so I will not now just not hold you any longer because this is just the exercise 3 is just short, and I will give you time to prepare your materials, prepare your the the things that you have to do much better if you could conduct um, today or maybe this week, ganun, so that you can really have a much time to prepare and also think how to do it. Okay, and then before I leave you, I will just share to you what Paracelsus said. So I think you just encounter this philosopher and this philosopher scientist that who said that poison is in everything and no thing is without poison. So the dosage makes it either a poison or a remedy. So too much of this food, too much of this chemical will make it poisonous, or less of this kind of thing, or less of this kind of vitamins, especially um, vitamin A, or yeah, less of this kind of vitamin A in our body um, be a, will have toxic effect in our body. So the dose makes it either a poison or a remedy. So class, always have in moderation in all foods that you are eating, especially now that you are at home, and especially that um, the things are overwhelming and you tend to stress eat, ganon. So always eat in moderation or drink in moderation if you are um, fond of drinking mga alcoholic drinks or such juices, ganon. So always eat and drink in moderation, class. So if there are no questions, class, or clarifications, so
Sa quiz, ma'am ba?
yes, yes, Schneider,
Ah, pila ka days, ma'am, na mo makakuan?
you have at least
Deadline nila.
24 hours to answer it.
Okay, ma'am.
So by 12 noon, I will post it in the VSUE and it will just open. And then tomorrow at 12 noon, so it will close. But there are specific time also that will you will just answer. So, mga na siya, mga 20 minutes, 30 minutes, ganon. So, the quiz is just easy, class. So, that's okay only. Okay, so maybe for the next week, class, I will not meet you because I will give you time also to really conduct also your experiment. So, conduct class. So, you have more time to conduct your experiment. So, this is just easy, but I will just give you an ample time because dili lang kay kaniman ang inyong subject. Okay? So, do you have any other questions before I uh, dismiss you?
And so far, ma'am.
And so far, okay. So before I dismiss you, I would like to request to open your camera and let's have a photo op. How about the others? Can you? Okay, so again, I will not take a photo, class. Okay, 1, 2, 3, smile. Okay, again, so Zero said, 1, 2, 3, smile. Okay, thank you so much, class, for attending. Bye-bye. God
Thank you, ma'am.
Thank you, ma'am.
Thank you very much,
Thank you,
ma'am.
Bye, ma'am. Thank you.
Bye.